So in the second part of this session, we're going to look at body fluids. Okay, so when we're thinking about body fluids, if we're thinking textbook wise in a 70-kilogram male, there's about 42 litres of water. And just looking at this little image, which is nice and clear for you, 2/3 of that is inside the cell there, and 1/3 is outside the cell. And outside the cell, you have two little areas. So you've got the blood, or the plasma, which is the water phase of the blood, and then you've got the interstitial space. So that is the fluid that surrounds your tissues and bathes your cells. And basically, you need to get nutrients from the blood vessels into the interstitial space and then into the cells. And then obviously the waste products have to go back to the blood so we can remove them. So like I said, of that 14, so if we calculate it, 42 litres, 2/3 is 28 litres. That's what's inside the cells, 14 litres outside the cells. Of that, 3 litres of plasma with another 2 litres worth of red blood cells. So you have roughly 5 litres of blood. 3 litres, like I said, is liquid. 2 litres is cells, which means 11 litres is your interstitial fluid, the stuff that surrounds your cells themselves, bathes your cells. The other important concepts you need to understand, and my chemistry is notoriously rubbish, is what is molarity, which is the number of molecules in a solution measured as moles per liter or millimoles or micromoles, and osmolarity, which is milliosmoles or osmoles per liter. Key difference really is that osmolarity is the number of particles in a solution as opposed to the number of molecules in a solution. I think that's the key thing you need to understand. And the number of particles, if you think about it, the molarity and the osmolarity of glucose is the same. So if you've got 5 millimoles per liter of glucose in your blood, you also have 5 milliosmoles of glucose in your blood. And that's because the glucose doesn't kind of break apart or dissociate in solution, whereas sodium chloride does. So you will have sodium chloride in your blood roughly 140 millimoles per liter, and the chloride bit is roughly in our blood about 105 millimoles per liter. So it's kind of, you think of sodium chloride as salt in a solid, you put it, use table salt, but actually in solution, they go apart into sodium and chloride ions. The other key thing to think about is that the osmolarity in your cells, in your interstitial space, and in your blood vessels is the same. It's 290 milliosmoles per liter. So everything added up, all the particles added up, makes the same concentration, same osmolarity, which means there's no net movement of water between them, because that's what osmosis is, the movement of water. But what is inside the cells compared to what is outside the cells is very different in terms of their competition. So the amount of sodiums and the amount of potassiums and the amount of chlorides is very, very different between them. So before we talk about how the fluid and therefore the nutrients get from our blood vessels to our cells, just need to kind of remind ourselves what each blood vessel does. So we know that the main blood vessels coming out of our heart are nice big arteries. So we're thinking about the aorta, in particular coming from the left ventricle. And our arteries come down, they break off into or split apart into arterioles, okay? Then come into our capillaries, and it's at the level of our capillaries where we get the exchange of nutrients and fluids before coming into our venules and then back to our veins, which goes back to the heart. The key things are that arteries have got big, thick walls, and that's because they're having to deal with very high pressures and they're delivering blood to our tissues. The veins are very low pressure and they return blood to the heart, and especially in the limbs, some of these veins have got valves to ensure one-way transfer. And the arterioles, which I've just shown here, these are the things that come from our arteries and deliver blood to the capillaries. And the key thing about these is they vasodilate and vasoconstrict, and therefore can alter the amount of blood coming to the capillaries and therefore the tissues that the capillaries are within. So if you look at these arterioles, if they vasoconstrict, just have a quick thought, is more or less blood going to get to that tissue, or to these capillaries? So if these ones vasoconstrict, less blood gets through, less blood gets to the capillaries. If they vasodilate, more blood gets to the capillaries and therefore more gets to the tissues. So that's how you alter the amount of blood that gets to your tissues. You vasodilate or you vasoconstrict, in particular your arterioles. When we get to the level of the capillaries, these have got very, very thin walls, and that allows fluid and small molecules to move between the blood and the tissues. And like I said, that's how we deliver nutrients to our tissues, is through these capillaries. So how do we get there? Well, we've already said that process of getting through the capillaries is called filtration, and that's purely because of pressure changes. So here's your blood flow, or your hydrostatic blood pressure coming from your arteries to your arterioles, and then we've come to our capillary here. And so what you have is you have hydrostatic pressure, and that forces fluid from the capillaries into the interstitial space. And remember, this interstitial space is surrounding our cells. Really important to note that in health, we're not really getting any proteins going through there because the filtration unit is too small. And obviously we're not getting any cells going through there either. So therefore, what's coming through here is our sodiums, our potassiums, our glucoses, amino acids. But what it isn't coming through here is proteins and cells. Okay, so that's the key thing. So your blood and interstitial fluid both contain all of these molecules, but the interstitial fluid contains much less protein than in the plasma. So a review question for you. I'd like you to read this question, have a think about what the answer is, and then we'll go through it. Okay, a student stands still for several hours during a long lab class. By the end of the day, their feet appear slightly swollen. Where does fluid normally move from the blood into the tissues? So, nice, easy question for you. Hopefully, you all have said C, capillaries, because that's the only place in health where we're getting fluid moving from the blood and into the tissues, because that's where our kind of filter is. Next question. Again, press pause. Okay, a 32-year-old office worker notices mild swelling in their feet after standing for several hours. This occurs because fluid moves from the blood into the surrounding tissues. Which pressure mainly causes this movement of fluid out of capillaries? So again, hopefully what you said on this one is increased capillary hydrostatic pressure. So the pressure within the capillaries forcing the fluid from the blood into the interstitial space and therefore delivering nutrients towards the cells and into the tissues. So remembering what's happened at this point, we've come in, our blood's come into the capillaries at the arteriole end, and fluid has left those capillaries. So we've lost fluid from our blood vessels. So this is what's happened. Blood's come through, fluid has passed through here, but cells and proteins haven't. So as fluid is lost here, but proteins haven't been moved through, as we go along and more fluid passes out into the interstitial fluid, the protein concentration within this capillary becomes greater and greater and greater, until at nearer the venous end, that protein concentration is now quite high. And so water now flows by osmosis from the low protein concentration in the interstitial fluid to the higher protein concentration within the plasma. And so it's doing it by osmosis. So water movement by osmosis. And the dissolved electrolytes, your sodiums, your potassiums, your CO2s and your oxygens, all come back because they're pulled along in that fluid. And this pressure, due to the protein pulling force, is called oncotic pressure. Okay, so it's a protein pulling force. So in other words, blood comes to the capillaries, fluid passes into the tissues to deliver nutrients to the cells, and then the fluid comes back at the venous end of the capillaries to maintain our blood volume. Okay, so our next question. Press pause. So proteins in the blood help to keep water inside blood vessels. What is the main effect of blood proteins on water movement? So A, B, C, or D? Hopefully you said C, they pull water into the capillaries from the interstitial space. So that's at the venous end. And what follows or what's dissolved in that water comes with it. So your dissolved CO2s, your bicarbonates, your glucoses, your CO2s, oxygens, whatever else it might be. So we just said that the fluid comes back into the capillaries because of that oncotic pressure, the pulling force of proteins. But not all of it comes in. In fact, only 90% or so of it is actually returned back to the plasma through this mechanism. The remaining 10% kind of sits there, sitting in our tissues. And it has to get, therefore, back into our circulation through a different route. And it does this through the lymphatic system. So that remaining 10% comes back through our lymphatics. So here's a little summary diagram. So here's the arterial end of our capillary. Blood coming in, high pressure, so bulk flow. Fluid leaves the capillary into our interstitial fluid. As the fluid has left and proteins haven't come this way, the protein concentration increases, and osmosis, so water movement, is dragged from a low protein concentration to a high protein concentration to make it the same on both sides. Okay, so that's your oncotic pressure, and that helps to maintain blood volume. But about 10% of it doesn't come back in this way, and it drains through our lymphatics and enters the blood elsewhere. So, key summary. Fluid moves out of the capillaries due to hydrostatic pressure, moves back in due to the pulling force of proteins via oncotic pressure, and the excess is returned to the body via the lymphatic system. So, another question for you to think about. Press pause. A 55-year-old man with chronic liver disease develops swollen ankles. Blood tests show low levels of plasma proteins. What's the most likely reason for fluid remaining in the tissue? So hopefully you've said reduced reabsorption of fluid at the venous end of capillaries, so option D, and that is because the plasma protein level is lower than normal, so therefore the oncotic pressure is lower than normal, so therefore not as much fluid is being reabsorbed back into the capillaries because there isn't as much protein pulling force, which means that fluid stays in the tissues, leading to edema. So that's the swollen ankles. Next question, press pause again. Okay. During a physiology experiment, capillaries observe where there is no net movement of fluid between the blood and the surrounding tissue. Based on today, so what we just covered, which explanation best accounts for this observation? So A, B, C, or D? So hopefully you said C. This is where the capillary hydrostatic pressure equals the plasma oncotic pressure. In other words, the pressure, the hydrostatic pressure in the capillary, forcing fluid one day, is equal to the pulling force of proteins pulling fluid back the other way. So while there's movement, there's no net movement. Last question, I think. Not all fluid that leaves capillaries returns directly to the blood. What happens to the small amount of excess fluid in tissues? This is a really nice, easy one. Hopefully you've said A, it enters the lymphatic system. It's about 10%. Okay, thank you very much. We are done.